Good morning. Welcome to my third video in Grow Your Maths and Science Like a Superhero. In the background, if you can see, this is Lord's Cricket Ground where I live. I live in London. Welcome to everyone from around the world. Asger, I hope we will be doing some more on plant biology next week. So today's one we're going to go into what DNA coding is, genes and chromosomes. We're going to explore what the central dogma of molecular biology is, which is how proteins are made from DNA, which is the programming code of humans and plants. So we're going to start with nucleic acids. What are these? So these are a chemical. And there are two types, DNA and RNA. So this is quite a long story. So DNA is deoxyribonucleic acid and RNA is ribonucleic acid. And if you can see on the slides, we're going to have a quick look at the chemical structure. So the chemical structure you can see underneath is a sugar, phosphate, and a nitrogenous group. So this is a particular one is a pentose sugar, pentose being 5. So you've got the 5 carbon. I'm just going to show you exactly what it looks like. So, carbon here, carbon. That's why it's called a pentose sugar. So this is all the different bonds. And then we've got a phosphate group here, which is PO4. And we've got a nitrogenous base, so this contains nitrogen. So the 4 particular nitrogenous bases are adenine, thymine, guanine, and uracil. These are polymers made out of monomers called nucleotides. Nucleotides, as we just discussed, made up of a phosphate, pentose, and a nitrogen base. So if we move on to the next so we're going to consider how nucleotides are formed. So this is the chemistry part of DNA. So formation of a nucleotide. So we discussed the subunits of nucleotide being the phosphate, sugar, and nitrogenous base. This is quite a good illustration. It shows you how much nitrogen is the base. So we've got, let me go back. So we've got 1, 2, 3, 4, 5 nitrogens in this particular nitrogenous base, which is adenine. We'll call this an amine group, being NH2. So here we've got our pentose sugar. In a nutshell, what happens is what's called a condensation reaction, which is water is removed when the nucleotide is formed. So if we look here, we can see H2O, which comes out off in the reaction. And this forms what we call a nucleotide. So let's move on a little bit more. So we talked a little bit about RNA, the difference being RNA is ribonucleic acid. So what the difference chemically? So if we look here, This is what we call a hydroxyl group. So then you've got the difference being this has got oxygen, and this one doesn't. And that's the only difference between ribonucleic and deoxyribonucleic acid. We talked a little about the bases. They're divided into two categories, which are purines and pyrimidines. Let's have a little bit more of a closer look at the classification. Adenine and guanine are purines, cytosine, thymine, and uracil are pyrimidines. It's important to remember in DNA, adenine, thymine, guanine. So I'm giving the shorthand here. Whereas RNA only has these. So adenine has uracil instead of the thymine. Okay. So best, when I think about DNA, I think of it as a long rope which unwinds and winds when replication of DNA occurs. So DNA is made up of a polymer of units called deoxyribonucleotides. We'll discuss the structure of these in the previous couple of slides. Made up of two polynucleotide chains linked by hydrogen bonding. And as you can see, there are different bonds between each of the different base pairs. So there's 2 bonds between adenine and thymine, 2 hydrogen bonds, and 3 between cytosine and guanine. So each polynucleotide chain is made up of 4 of these nucleotides. So guanine, deoxyribose, phosphate, adenine, cytosine, and thymine. And these undergo what's called a polymerization reaction and arrange to form a polynucleotide unit. So on the left, you can see the pairs, and on the right, you can see how they coil in DNA and pair up. We discussed a little bit about condensation reactions when a water molecule is removed. And on here, it shows you a little bit more detail of what a polynucleotide chain is. So you can see nitrogen bases extend outwards from each sugar group. So base pairing, how does that work? In a nutshell, there's only certain bases that can pair with each other. So I'm just going to show you adenine always pairs with thymine. Guanine and cytosine. Thymine and adenine. And cytosine. Guanine. These are what's called complementary. Okay. It's important to remember that the different types of bonding, there's 2 hydrogen bonds between adenine and thymine, and 3 between guanine and cytosine. A little bit more on nucleic acids. So originally this was discovered by two guys called Watson and Crick. I think this was 1967. And it was originally named the Watson and Crick model. What's the function of DNA? It stores all the genetic information and conveys this information to all of the different cells and is contained in all the different cells in the body. Controls every single metabolic process and varies occasionally due to the ability to change itself and depending on what the code is. A little bit about DNA replication. What is replication? So it's a key cell process, meaning how we form 2 identical molecules from a mother DNA molecule. So 3 steps. 1 is unwinding it, 2 is assembling the base pairs, and 3 is formation of new strands. So this is quite a good diagram. You can see all the different base pairs here. So you've got all of these different adenine, thymine, guanine, cytosine, and so on. And you can basically see that this is unwound at this point here. So what happens in DNA replication? So we're going to talk a lot about enzymes in a lot of different settings and locations in the cell over the coming months. There's several key enzymes involved in DNA, and broadly these are categorized into these groups: ligases, primases, helicases, polymerases. Have a look at what different types there are of these. In this particular example, we're going to talk about DNA helicase. And a helicase is an unwinding enzyme. Remember, an enzyme is basically catalyzes a particular reaction, and they're all quite specific. We'll go a little bit more into proteins, types, conformations in another module. So here we can see DNA helicases catalyzes the breaking of hydrogen bonds. Remember, I said before, hydrogen bonds link the different nitrogenous bases here. So it exposes all these different ones. And so the different colors are probably something like this: guanine, cytosine. Then you've got the pair. So you can see these are the different bonds that are broken, exposing the different base pairs from one strand, so the new strand can be synthesized. So each strand now acts as a template for the new strands which are about to be formed. So assembling of new complementary bases. So what happens? 3 nucleotides in the new cytoplasm arrange in front of these exposed ones. So they slot in nicely here. Remember we said before, adenine thymine has 2 hydrogen bonds, whereas cytosine guanine has 3. And these are catalyzed by DNA polymerases into formation of the 2 new strands. So DNA ligase is basically the joining enzyme which facilitates the joining of the new strand. So the next part we're going to go through is RNA. So I've covered what the chemical structure of RNA is. RNA is a polymer. Its monomer is a ribose nucleotide. And I've also mentioned, instead of thymine, it's made of uracil, a particular uracil base. So it's still got uses the other 3 bases, which are guanine, cytosine, and also adenine. I'm just going to erase this out. Just a minute. So there's 4 types of nucleotides in RNA which undergo polymerization, and these combine forming phosphodiester bonds to form an RNA molecule. And exactly the same, it's called a condensation reaction. So let's put this into context in cells, which I sort of touched on in the last one of the early videos. Eukaryotes and prokaryotes, they all have DNA. So if we have a look at the differences between the two, you can see this is what we call an organelle. And in this organelle, you've got all of these different organs. So we've got mitochondria, ribosomes, nucleus, nucleolus here, lysosomes, and other organelles called endoplasmic reticulum. So in short, mitochondria are basically the energy factories. Ribosomes I call the protein factories. We're going to mention a bit about ribosomes shortly. So chromosomes exist as linear DNA, so i.e. straight within eukaryotic cells. Chromosomes are thread-like structures found in this part here, which is the nucleus. Let me just so just here is the programming center of every single cell, containing thousands of meters of DNA. And the DNA molecule is found wrapped around histones, which are proteins, which give DNA structure and support. Compare that to prokaryotic DNA, where you've got short circular DNA. It's not got any histones. And we refer to supercoiling, which is, if you think of a rope, if you twist a rope and it gradually gets coiled more and more and more. And that's how so much information is packed into the DNA code. So a little bit more, we're going to go into more about genes now. So what are genes? So a sequence of DNA bases which codes for a particular functional RNA unit or a polypeptide. And a gene exists at a particular position or locus in a DNA molecule, formed by a particular polypeptide. So the structure of genes and DNA. So we can see here, so you've got your big DNA rope supercoiled, and into packing units called histones, which are proteins associated with DNA. A particularly good diagram here, which shows how DNA is wound and packed into chromosomes. So remember what we said about a gene. A gene is a segment of DNA. So a little bit about introns and extrons. These are sections of DNA that do or do not code for amino acids. So how does DNA control activities in the cell? Well, it's all chemical reactions controlled by enzymes. So this is how DNA controls the cell's activities. So a little bit about protein synthesis. So in the nucleus, DNA unwinds and unzips as the hydrogen bonds break between bases break. This is what happens in replications. And then we have RNA nucleotides in the nucleus, which are only one strand. So we didn't mention that before. DNA is a double-stranded molecule. RNA is a one-stranded molecule. So what happens is, as DNA leaves the cell, so we talked a little bit about the processes, transcription, and translation. So in the cytoplasm of any cell, there are 3 amino acids and transfer RNA molecules. So it's important to remember, I mentioned before the ribosome. I call this the protein factory because it zooms along this, making a growing polypeptide chain of amino acids. And in a nutshell, that's exactly what happens in protein synthesis. So at one end of each transfer RNA molecule is a site to which an amino acid can bind. So this is the 5' and 3' end. And at the other end, you have 3 unpaired bases. So this particular base, each triplet, which I'm going to show you here, we have a particular name for them. And these are called anticodons. And each transfer RNA molecule binds with a particular amino acid under the control of a particular enzyme using energy called from ATP. And we're going to cover ATP structure and function in the next module. So a ribosome has two units, a large subunit and a small subunit. And these basically sit on the transfer RNA, recognizing its codon from the mRNA, binds to it through the by complementary base pairs, and then slots in an amino acid to the grown peptide chain. So a peptide is just a protein. Right, shrink that a little bit and move on to the next part. So this is a little bit more of how amino acids form. So in the cytoplasm, we have messenger RNA. So remember, messenger RNA, as it sounds, is a message from the DNA, which then attaches to a ribosome made up of ribosomal RNA. So we can see here how each subunit forms different amino acids. So the particular ones here it's making are, I'm going to come to the table of amino acids at the end. So we've got methionine, and we've got phenylalanine, and then down here it's making, we've got arginine. We're going to come to what the different code is right at the very end. A bit more about protein synthesis. So here you can see the different ribosome subunits, the messenger RNA and the transfer RNA. So the transfer RNA is moving along there. It uses an enzyme called a peptide transferase, and it goes along, and then you can see this new protein being made out of different polypeptide units and amino acids. So this is a good overview of what I've described so far. Transcription of combinations of these three nitrogenous base pairs: guanine, adenine, thymine, and cytosine. So here we go. These are all the different amino acids you can see here. So we've got alanine, cysteine, aspartate, so on, and these are the one letter codes. So in the news recently, we've seen something about a particular COVID mutation called N501Y. So basically this was a slight change in two amino acids. Asparagine and tyrosine. So those two amino acids were changed from the original sequence, and that's what one change in amino acid can cause. So if we move on slightly. So we're going to go a little bit more into mutations, and alteration of the sequence of bases in DNA can alter the structure of proteins, which I briefly mentioned. What are mutations? So mutations are a change in a particular part of the genetic material, which can obviously be transmitted to future generations or inherited. What causes them? So can be an error in replication or segregation of DNA. Chemicals can cause DNA mutation, or even UV and gamma rays. So obviously gamma rays, radiation, they physically damage DNA, break it down in cells. Temperature and pressure, high temperature, and mitosis, meiosis, so how cells are divided. So what exactly happens in a mutation? So we explained before that amino acids are made out of reading 3 of these. So what can happen is you can have a substitution, which causes a different amino acid to be made, an addition, so an extra amino acid. So here you see there's an extra adenine. Deletion, you can have one removed, or duplication, where you've got one particular nitrogenous base that's been duplicated, or inversion. So it looks a little bit like this. So this is insertion in particular chromosomes, translocation. So it's important to remember the sequence of amino acids is determined by the order of DNA bases in a gene. Therefore, one mutation in a gene can alter that amino acid sequence in the polypeptide. So a chemical change. Now, genetics tends to have a degenerate nature, which means that certain amino acids are coded for by more than one DNA triplet. I showed that on the last table. So it doesn't necessarily mean there will be a change, just that there could be. And some mutations don't actually alter the amino acid. These are called silent. So this is quite a good example. So the silent has no effect on the protein sequence. So you can see in this particular illustration here, this one was changed, but the same amino acid was made. Got missense, which is a particular amino acid substituted. So this example here, this one was substituted for this one. And you can see here, different amino acid made. So this one is proline, and this is threonine. And then a nonsense where the stop codon has been read. So I didn't go too much into that. We'll come to that another time, but basically there's a start and a stop codon, which are a coding sequence which tells the DNA or RNA when to stop making the proteins. So we're going to put this into context. So genetic disorders, there's quite a lot of them. So these are basically minor changes in the genes, DNA, which can cause any one of these. So we pick an example. Got one with cystic fibrosis, which is an error in a particular chloride channel. Sickle cell anemia is deformed blood cells, red blood cells rather, which aren't shaped the same as normal erythrocytes. There's plenty of others. So we've got muscular dystrophy, which is quite common, Huntington's disease here. So probably the ones that people will have heard more of. This one, hemophilia, clotting disorder. This is quite common. Sickle cell anemia. And there you go, we've got a couple of immune deficiency diseases caused by errors on this is chromosome 20. So remember there's 23 chromosomes in the human body, or 23 chromosome pairs making 46. Okay, so that brings me to the end of the section on DNA. So thank you very much for those of you that watched this. I hope you found it a useful explanation. Please do comment if there's anything you want to ask. It's been a pleasure. This is my first recording on DNA for this group, and I hope you'll enjoy it. See you next time. Bye.